Great, so I'm going to take us in a very different direction here, and if I had to define my talk in a sentence, it would be where big data, small data collide in order to make decisions about public health. I work on a particular pathogen, disease-causing microorganism called Legionella and You may have heard of it. There's been some really big outbreaks in New York City over the years, and in recent years it's gotten a lot more attention as it started to comprise a larger portion of waterborne disease outbreaks. So as it's gained notoriety and we've started to systematically look at some of these outbreaks, people have started to bring up new questions about how data science can inform kind of how we do this. And because risk analysis is really a trans science, it requires somebody to integrate data from a lot of different settings, ranging from clinical to laboratory to social science. And based on a lot of the work that's come out recently in terms of summarizing these problems, a lot of the problems are linked to the built environment. And that's really conducive to collecting a lot of data. So, when I work on water quality issues, a lot of times the regulatory purview ends at the property line, and that's when the responsibility of the homeowner begins. So when you talk about a pathogen like Legionella, you get exposed from inhaling mists that result from different fixtures in your home and then reach the lungs. In terms of how we can use data to inform regulatory decisions, the EPA, Environmental Protection Agency, operates on a maximum contaminant level type paradigm, where even though we have distributions of data coming in, people want it in practicality to be condensed to a single number to say, oh, if a concentration of something is above this, it's a problem, and if it's not, then we're okay. So where this becomes an issue is that if you look at all the guidance out there, that the people that have to make decisions about this and manage engineered systems and buildings, the estimates are all over the map as to what should be allowable in water in terms of concentrations of this particular pathogen. So where the data comes in terms of integrating different data streams is through this process, microbial risk analysis, where we identify a problem, look at relationships between what people are exposed to in the environment and some sort of response through quantitative function, integrate that with information about human behavior, and arrive at some sort of risk estimate and the drivers of the risk so we can manage them. An example would be in the clinical environment, some people will dose different organisms with types of bacteria and observe the response and fit a quantitative function to that. So these data are exceptionally sparse, so probably the opposite of the problem that a lot of people in biodiversity have in terms of having to use small data sets and use sparse data analysis techniques to integrate that, and then also maybe use systematic methodology in order to review a lot of literature in order to find these data sets, so in terms of systematic review and computational methods to do that. Then we also look at laboratory data, so for example, a data set generated from American Water in terms of measuring concentrations of pathogens in the lab with different computational techniques, in addition to instrumentation. And I'll come back to that and circle back to kind of microbial diversity and how that's important in this process. In terms of doing this, I'll sort of skip over some of the details of the risk analysis modeling that we do, but by integrating these data streams, I can get to sort of risk estimates for different types of fixtures in the built environment, and then aggregate exposures to water sprays from those fixtures. And based on different criteria that we set a priori, work backwards and figure out what an allowable concentration in water would be to try and get at that MCL process, that allowable concentration process. So I've done that here to show the differences in different water fixtures and how your exposure risks might vary. Over some of this, but if you simulate this over a whole hypothetical series of concentrations, we can work backwards at what the allowable concentrations would be for different target risk benchmarks. And I've done that here, so you can see that this does vary depending on which kind of target benchmark you choose, which type of water fixture you're concerned with. But I guess where this is interesting in terms of where other data streams come into play, microbial diversity in the built environment is a very hot topic in terms of research. And as we start to get more high throughput sequencing information, trying to integrate that into a risk and decision analysis framework is not always very easy. So that's something that I'm kind of looking to partner with others working in that sphere on, and how to integrate that into more of like a decision analysis type of model. And in terms of what I have here, microbial communities interact with each other, and I'm trying to translate these data streams we get from sequencing into quantitative rates of either transfer or interaction between different microorganisms that have symbiotic relationships with the pathogens that can make us sick. And Legionella is very well known for having symbiotic relationships with certain other organisms in microbial communities. In terms of measuring pathogens in the environment, this is an example, and this is my last slide of some of the types of data that are generated from high throughput efforts in molecular biology labs up at the top right corner. And you know, the response when water treatment or other professional gets this is, cool, what do I do with that? So you know, we get a lot of information from these types of things, and the integration in terms of what should be done in the policy or management context is not always there. So that's really where I'm looking to focus and collaborate with others. So thanks for having me. I'll happy to go to more detail.